This lecture will be about enzyme kinetics primarily, and at the end we'll talk a little bit about mechanisms and some regulation of enzymes. But primarily, I think it'll be um, mostly the lecture will focus mostly on the kinetic aspects of enzyme reactions. And these slides were provided to me once when I had to teach the end of uh, this class at DU, and I really didn't have any time to prepare. Um, I don't know if I like them a whole lot, but I did like them more than the book slides, so I decided to use them. That's why they look a little bit different than the previous lectures. Um, these slides go through the derivation of the Michaelis-Menten equations in as well as the different types of inhibitions you can get with that. And those derivations are a little bit outside of our class, the scope of our class, but I think they might help you to see them. But I'm not going to be asking you to derive those on an exam. So you really, I'll try to focus in and tell you the the equations that you need to remember. Um, but sometimes I think it helps you to sort of remember the Michaelis-Menten equation. If you can sort of see where it's coming from, it might help a little bit instead of just trying to memorize the equation on its own. Okay, this is kind of this title slide here is really just what we're going to be the outline of the lecture, what we're going to be kind of going through. Um, so as a review, if we had this basic generic equation, A plus B going to C plus D, we can write a equilibrium constant for that, right? And it'd be products divided by reactants, C times D divided by A times B. Okay. Um, we can write a equation for the delta G of that as well. Okay, and the delta G, we can write the equation delta G equals delta G naught plus RT times ln of the KEQ. Okay, and that equation you'll see a lot more of in metabolism. When we cover metabolism, you usually talk a lot more about thermodynamics. Delta G naught is the delta G at standard state. So the delta G at standard state doesn't necessarily mean anything about what the delta G is in cellular conditions. And you'll see in metabolism that you have some reactions that do have an unfavorable delta G at standard state, and they become favorable because you can adjust these concentrations inside a cell to make them favorable. But as this is really sort of as an aside, because delta G and KEQ uh, are really the driving force of a reaction, but they don't say anything about the rate of the reaction or the kinetics of the reaction. Okay. In reminding ourselves, okay, enzymes are catalysts, right? They make reactions go faster. Um, Most enzymes, almost all that are known, are proteins. There's an exception of that are ribozymes that are actually um, a mixture of proteins and RNA, uh, but they still have a protein component. The RNA part is what is the catalyst, though. Okay. They bind molecules, reactants very specifically, and they involve intermolecular forces in those binding. Uh, and what they do is, as we talked about last time, they stabilize transition states. They lower that transition state energy. Right, and this is a figure I did show last time, right? And just as highlighted here. These aren't very important to memorize or remember, but it just shows you some of the very big enhancements you can get with enzymes, right? So carboxypeptidase A, that normal half-life would take 7 years. The uncatalyzed rate is shown here. Catalyzed rate is much faster, right? 1.9 times 10 to the 11th. And then carbonic anhydrase, which we've mentioned previously, and we'll see some more of this at the end of class, an example of the mechanism of this enzyme. But it has, that reaction is a fairly quick reaction, has a half-life of 5 seconds without an enzyme. But the catalyzed rate is 1,000,000 with the enzyme, right? So that's, a very huge rate, 1,000,000 a turnover number, okay? So in transition state theory, the enzymes, both enzymes and temperature, help increase the rate of a reaction. And here we have an equation, Boltzmann equation for the rate constant. Okay, so this is the rate constant, and Boltzmann constant times temperature divided by Planck's constant, which you might remember from Gen Chem. And then you have the e to the negative delta G of the transition state divided by RT. So temperature factors in a couple times here. And so does the transition state energy. So temperature works in both places above, right? So raising the temperature increases this factor, and you want to lower this exponent here since it's negative. So adding a higher temperature in that the bottom of this fraction here will lower this exponent. Okay, so that in both cases, raising the temperature there makes the rate higher. What an enzyme does is it lowers that transition state energy as well. So it makes this term lower, which makes the overall exponent here lower, this lower, which makes this higher, a higher term. Okay. Enzymes don't alter the laws of thermodynamics, and that means they don't alter the delta G of the reaction. They also do not alter the equilibrium constant. Okay. So if the equilibrium concentration of B, in this example, A going to B, we have the reverse rate and the forward rate, so we have rates of both of those directions. Okay, if they're at equilibrium, we can write an equilibrium constant for that. It would be B over A, and that would equal to the rate of the forward reaction over the rate of the reverse reaction, right? And we can say if equilibrium constant is 100 for an uncatalyzed reaction, the enzyme catalyzed reaction, that equilibrium constant is still going to be 100. Right, the enzyme doesn't do anything about the equilibrium. It raises the rates of reaction, but it raises the forward rate, and it also raises the reverse rate of the reaction by the same amount. Okay, so that's something that's very important to remember about enzymes. They speed up the forward reaction. They also speed up the reverse reaction. So they don't affect the equilibrium, but they do accelerate how quickly the equilibrium is attained. And again, delta G of the transition state does not enter into the final delta G of the reaction. So delta G of the products and the reactants are not changed. So the overall delta G isn't changed, but the transition state delta G is lowered. And we showed these last time, so I won't really go through it too much. Right? This would be a normal reaction, or you have a large transition state, and I should say an uncatalyzed reaction. Okay. equilibria are linked to the standard free energy change. Reaction rates are linked to the activation energy. Okay, so equilibrium are linked to the delta G of this reaction. The rate of the reaction is governed by this transition state. So when we catalyze this reaction, we get the blue curve, right? And that lowers, you have this lowering of the delta G of the transition state. Okay, and that makes for a increased rate going from substrates to products. But it also, as we mentioned, if we're talking about the reverse reaction, it's also going to speed up the reverse reaction of products going to the substrate. Okay. The rate limiting step is the we'll mention this again, but there's the first step is the binding of substrate to the enzyme, and then the transformation of that substrate to a product when it's on the enzyme. This is the rate limiting step. Okay, and enzymes, again, don't affect reaction equilibria, they just affect the rate. Okay, and this is, we mentioned this last time, this idea of induced fit, and we talked about that idealized case of stickase with the enzyme. The shape of that binding site is actually complementary to the transition state, and that's how they act to lower that transition state energy. Just a brief kind of review of kinetics. If we have a very simple reaction, substrate going to product, we can talk about the velocity of that reaction, the rate of that reaction, symbolized by the V. That's going to be equal to the rate constant, small k. Okay, don't get that confused with the equilibrium constants that we also talk about that are capital K's. Okay, small k is a rate constant. Okay, so small k times the substrate concentration, that will give us the rate of this reaction. Okay, this is unimolecular or first order. If we have a bimolecular reaction or second order, okay, this term becomes, for the rate, would be equal to rate constant times concentration of substrate 1 times concentration of substrate 2. So we multiply in by the second molecule there. And then you can also have a third order reaction, okay, which we won't really talk about in this class, but you just add, you know, the third substrate in there. Okay. In enzyme kinetics, it almost, I would say always, we're going to be dealing with Michaelis-Menten kinetics. And the Michaelis-Menten equation, we're going to sort of act to derive it here. The reaction that we're looking at in Michaelis-Menten kinetics is the enzyme catalyzed reaction. There's a few steps. So the first one is enzyme and substrate, or our you can think of that as sort of the bimolecular reaction. They have to come together and form an enzyme substrate complex. Right? And it turns out this is a fast step, and the dissociation of that substrate off the enzyme is also fast, and this is in an equilibrium. Okay. And then the rate limiting step of this is a two-step process, right? First we bind our substrate to the enzyme, and then that substrate is then converted to a product and it leaves the enzyme active site. Okay. So the first step is fast and it's an equilibrium. The second step is the slow step, the rate limiting step. Okay. So we have this forward reaction here is K1, the reverse of the dissociation of that substrate is K minus 1, and then our K2 is this second step that's only one direction. Okay. And the assumptions that we make, enzyme and substrate react rapidly to form that complex, single substrate and single enzyme substrate are involved. they break down directly to enzyme plus product. Okay, so we don't have two enzyme substrate complex being needed. It's just one of these complexes that lead to the product formation. Enzyme substrate and enzyme substrate complex, there are equilibrium. Enzyme substrate dissociation is faster than enzyme substrate conversion to enzyme and product. Okay, that's why this is the rate limiting step. And substrate concentration in general is much greater than enzyme concentration, and therefore substrate concentration is also much greater than enzyme substrate complex concentration. So the formation of this enzyme substrate complex doesn't alter this substrate concentration. So those are really some assumptions we make in this. Okay. We're going to show this plot, and we'll be coming back to this plot quite a bit. And what this plot is we're plotting what's known as a initial velocity, or V naught. So that's the initial rate of reaction. And the units are in micromoles per minute here on this graph. And we're plotting that versus the substrate concentration. So in practice, what you would do is you would take your enzyme and you have some way of measuring its activity, how fast the rate is going in the reaction. And you would have your enzyme, same amount of enzyme, and you would just add each time you would add a little bit higher concentration of your substrate, and then you'd measure your initial velocity. And when you do that, you get a plot that looks like this, sort of hyperbolic. Okay. This line here is showing you what the initial velocity is. What you're doing is you're taking that velocity right away. So this is kind of like a tangent line to what the initial velocity would be down here. Okay, so that's what if it continued, the velocity continued. If we're just plotting the rate of the reaction and following the reaction, it looks something like that. But in practice, as you increase the substrate, you get saturation of that active site, and it starts to slow down at higher product concentrations. And it reaches an asymptote. Okay, and up here, that's really the highest this graph will ever go. And we say that is the V max. Okay, so this, when it sort of goes horizontal here, this graph goes horizontal, whatever value that is the V max. And sorry, you can't read that. We'll see that equation again shortly. The other point I'll make now, we'll be talking about this more though. So we have a V max up here at the asymptote. And that's at a particular value of V naught. If you look at half of that value, so 1/2 the V max, and you follow it over to our curve, whatever value that occurs at the half V max, the substrate concentration there, we call that the Km, right? And that's a capital K. That's the Michaelis-Menten constant for a particular enzyme. Okay, so that'll be important to remember. That's on a graph like this, that's how you find that Km. Okay. Right, so we when we do Michaelis-Menten and we derive the Michaelis-Menten equation, we're assuming steady state, the steady state assumption. So the steady state assumption is really saying that the amount of enzyme substrate complex is at a steady state. It's not changing. And when that happens, the rate of our reaction is simply this part, the second part. So V naught equals K2 times this enzyme substrate complex concentration. And then this, I'll go through the derivation here pretty quickly. If you're interested in it, you can, look through the slides in a little bit more detail. But this, again, you don't really need to know this for this class to do well in the class. But you might want to know this if you're doing enzyme kinetics at all. Really, I don't even know in practice if you need to know all this for enzyme kinetics. But some people might find it just kind of helpful to know. All right. So we can talk about the total enzyme concentration that equals the free enzyme plus the enzyme substrate bound. Okay, if we rearrange this equation in terms of free enzyme, it'd be the total enzyme, so that's how much we added initially, minus this enzyme substrate complex. That's what we'd use to get our total enzyme. So in the first step, you find the rate of your complex formation and your rate of complex breakdown. Both of those would be given here. Then you can expand and factor these out. So then you apply the steady state assumption, right? And the steady state assumption says the rate of the formation of this complex equals the rate of the breakdown of that complex. So you take your rate equations for the two situations, the formation and the breakdown, you set them equal to each other. Okay, that's the steady state assumption. Okay, when we expand and we factor these out, the results are shown there. Okay, and then the next step is solving for that enzyme substrate concentration. So when we do that, we get enzyme substrate concentration is equal to this. Lots of different rate constants, and we have this in terms of the total enzyme concentration and the substrate concentrations. Okay. This, again, not really that important to us yet. Okay, we divide the top and bottom by K1. All right, and when we do that, this is what we're left with. Okay, and now we define K2 plus K minus 1 over K1. We define that as the Km, the Michaelis-Menten constant. So that whole part of this term simplifies to Km. And we're left with the enzyme substrate concentration equals total enzyme times substrate divided by Km plus substrate. Okay. And when we express this in terms of a V naught, remember we mentioned V naught equals K2 times ES. So we multiply, what we do is we multiply both sides of this by K2, so we get it in terms of V naught. So that equals K2 times total enzyme times substrate divided by Km plus substrate. Okay. K2 times total enzyme concentration turns out to be the fastest rate you can have, which is the Vmax. Okay, so this term simplifies further to Vmax, and what we get finally is the Michaelis-Menten equation. So V naught, or reaction velocity, equals Vmax times substrate concentration divided by Km plus substrate concentration. So this right here is really the important part to remember. We can look at Michaelis-Menten in certain special cases. So here's our Michaelis-Menten equation. Okay. What is the substrate concentration when we're at half of the maximum rate? So if we divide by 2, okay, and we read that off the graph of rate versus substrate, okay, what we get is 1/2 equals substrate divided by Km plus substrate. Okay, and Km equals that substrate concentration when V naught is 1/2 of V max, right? So this is really the reasoning behind how we can read the Km value off of a graph if we just look at half of V naught equals half of V max. Okay. Right. Another special case, or special cases, depends on the concentrations relative to each other. Or I should say the substrate concentration relative to the Km. Okay. So V naught at very low substrate concentrations, Km will be much, much higher than the substrate concentration. And you can sort of ignore, so in the denominator here, if Km is much higher than substrate concentration, adding that concentration to the Km isn't going to do much. So you can really kind of ignore that. And this would simplify to Vmax times substrate over Km. Okay. And the opposite is a situation where you have a very high concentration of substrate. Right? You have a high concentration of substrate. Km in the denominator is really gonna, you can sort of ignore that. Okay, and in this case, right, the substrate concentrations cancel each other out, and V naught is just the V max. So really, when you're in that, we saw that on the plot, when you're at very high substrate concentrations, you reach a maximum, and that is equal to the V max right up here. Okay, so as substrate concentration goes to infinity, right, it goes, really, really high, V naught is equal to V max. Okay, and when S equals Km, or I should say when V naught is half of V max, that substrate concentration is Km. Right, and so in these cases, this would be the case of very low substrate concentration. V naught is equal to V max times substrate concentration divided by Km. And then at this part of the graph way over here, V naught is equal to V max. Everywhere else along this line, right, we have the V naught equals V max times substrate concentration divided by Km plus substrate concentration. Right, so those, are the situations, really the 4 situations you should kind of be aware of on this plot. Right, very low substrate concentrations, you have this relationship. Very high substrate concentrations, you have this relationship. There's a very special case when you're at half of the Vmax, that's the Km. And then everywhere else is governed by the general Michaelis-Menten equation. Right. We can rearrange the Michaelis-Menten equation, and this is where we get what's known as the Lineweaver-Burk equation. And for you do this to get a linear line that you can apply the sort of y equals mx plus b and be able to read off from the plot important parameters. Okay. And to derive this, if you have our Michaelis-Menten equation, that's where we're starting from. If you invert both sides, okay, so you have 1 over V naught, and you can basically flip these around. Okay, then if you expand this, right, so Km plus substrate concentration, Km over V max times substrate plus substrate divided by V max plus substrate, right, equals 1 over V naught. You can cancel the substrate concentrations here and get 1 over V max. And this is the Lineweaver-Burk equation. Okay, 1 over V naught, that's our y value, which would be our y-axis on our graph. Km divided by Vmax, Vmax times substrate. That'd be our x-axis. and plus 1 over Vmax turns out to be our y-intercept. So in this case, when we plot 1 over V naught is our y-axis, and 1 over S is our x-axis, our slope becomes Km divided by Vmax, and the intercept again is 1 over Vmax. Okay, so we have the y-intercept is 1 over Vmax, slope is Km over Vmax, and the x-axis is negative 1 over Km. Okay, so you can read off just on one of these plots, you can determine the Km and the Vmax. Okay, all enzymes that have this hyperbolic dependence of their initial velocity V naught on substrate concentration follow Michaelis-Menten kinetics. And as a practical rule, Km equals that substrate concentration when V naught is 1/2 Vmax. Our Km value is equal to those rate constants, K2 plus K minus 1. So that's the rate of formation of our product plus the rate of dissociation of our substrate divided by the rate of binding of our substrate. That's equals to our equilibrium constant, the Km. When the conversion rate to product is small, when K2, that means K2 is much smaller than K minus 1, this simplifies into K minus 1 over K1, and that is the dissociation constant, or KD, capital KD, that we sort of talked about last time. So that's another special case where you can, in a sense, you can approximate the Km value by finding a KD value. Examples of some Km values for different enzymes. Not important to memorize these, just gives you an example of the scale that we're looking at for some of these. And the higher the Km value is an more indication of how tightly bound substrates are to enzymes. So if we go back, the higher this value, I guess the opposite would really be true. If your rates here are higher, your product formation or your dissociation rate, that would give you if those are higher, you're going to get a higher Km value. Right, so the lower the Km value, or I really shouldn't say it that way, the stronger the binding of a substrate to an enzyme would lead to a lower Km value. A weaker binding substrate to an enzyme would give you a higher Km value, right? But the Km value by itself doesn't necessarily tell you anything about the binding of the substrate to the enzyme, because you do have that K2 term in there as well, which is how fast that enzyme substrate complex makes the product. So that also factors in there. Okay. Right. A definition to know is turnover number, and this is telling you how fast an enzyme is. So if we look at our Michaelis-Menten equation, let me get my laser pointer again. Right, our Michaelis-Menten equation. Right, and we can write out our Vmax. We have that relation between Vmax, it equals our K2 value, times our total enzyme concentration. Right. So our what's known as Kcat, or turnover number, that's the number of substrate molecules converted to product per unit time on a single enzyme molecule when the enzyme is saturated with substrate. So Kcat is more general rate constant, or first order rate constant, reflecting just the rate limiting step. Okay, so we can write, Vmax equals K2 times that enzyme. We can write this as K2 equals Kcat, right, in the situation where we have a substrate that is saturated. Right, and we can plug that Kcat times enzyme concentration into our Michaelis-Menten equation in place of Vmax. And so some Kcat values for particular enzymes, the highest one on this list is Catalyzed, which is probably the fastest enzyme known that I'm aware of. Okay, that's 40 million per second. So 40 million substrate molecules are converted to product per second on one of these enzymes, which is just crazy. Another definition to note is what's known as the specificity constant, and that's Kcat divided by Km, right? So remember, Kcat is a rate constant. Km is the Michaelis-Menten equilibrium constant. Okay, and what this does is it sort of combines both information about binding, which you sort of get from the Michaelis-Menten constant, and rate from Kcat. Okay. So when we look at our V0 in terms of our Kcat, when S is very, very small compared to Km, we can kind of ignore that, right? And then our V0 becomes Kcat over Km times total enzyme times substrate. Right? And this Kcat over Km is becomes a second order rate constant. And we can see examples of some of these here, which we'll sort of look into. This specificity constant, Kcat over Km, 10 to the 8th to 10 to the 9th per mole per second is really kind of the maximum you can have for a diffusion controlled reaction. That is where your substrates have to diffuse in solution and find their way to the enzyme and then be released. Okay. And some of these enzymes get very close to that limit. This acetylcholine esterase, gets really close to that, as well as some of these other ones. Right? We'll see an example of this later, and in carbonic anhydrase again later. Right. Saturation kinetics. What does that mean? Well, saturating the enzyme with substrate, right? That's getting higher substrate concentration. You're doing that, you're going to find Vmax, right? So you saturate with substrate, right? That pushes the equilibrium further to the enzyme substrate complex. Okay, that really becomes known as steady state, right? And the overall rate of the overall rate is proportional to the concentration of your enzyme substrate complex. So let's look at an example calculation. Right, so the rate of an enzyme catalyzed reaction is 35 micromole per minute at a substrate concentration of 10 to the 4th molar, 10 to the minus 4th molar. Km for this substrate is 2 times 10 to the minus 5th molar. Calculate the rate where substrate concentration equals 2 times 10 to the minus 6. So how would you solve this problem? Well, you're given a rate, and you're given the substrate concentration that's at, and you're given a Km value. So if you look at your Michaelis-Menten equation, we're given a rate, we're given substrate concentrations, and we're given Km. The only thing we're not given is this Vmax. So we can use all that information to calculate a Vmax number. So when we do that, we can get, with these numbers, we get 42 micromole per minute for our Vmax. Okay. So now the next step is calculating the rate where substrate concentration is 2 times 10 to the minus 6 micromoles per minute. Okay. So we found our Vmax is 42, so we can plug that in. We then plug in our new substrate concentration, and then we use the same Km value, and we use that to calculate the V naught, and in this case it's 3.8 micromoles per minute. So you can use this to actually predict rates of enzymes under specific sets of conditions, that is, specific substrate concentrations. Okay. If we look at in a time course, graphically, if we look at this, we're starting with a certain substrate concentration, okay, and at that starting point you don't have any product, that's shown here in the green. You have a certain amount of enzyme. Usually the enzyme concentration is going to be lower than your substrate concentration because it's only needed in a catalytic amount. Okay. So at the starting point, we have some enzyme, some substrate. What happens is your enzyme and substrate concentrations will start to go down, okay, and your enzyme substrate concentration complex will go up. And it'll do that, and once those get formed, that enzyme substrate gets formed, you can start forming some product. Okay, right there you're starting to convert your substrate to product. Okay, these become, the substrate starts to decrease linearly, the product increases linearly, and what happens here, right, with the enzyme substrate complex and the enzyme concentration, they reach what's known as steady state, so they're not really changing very much at all. Okay, this is what we talk about when we talk about steady state kinetics. That's the enzyme substrate concentration isn't changing. Okay, but your substrate concentration sure is, it's decreasing, and your product it keeps increasing, but the enzyme substrate complex itself is not changing. Okay, so just remember that for steady state. It's talking about that enzyme substrate complex. Right. Now we're going to talk about inhibition of enzyme reactions and how that relates to the kinetics. And there's 3 types of inhibition. Reversible inhibition. First, we'll talk about what's known as competitive inhibition. That's where the inhibitor competes for the active site with the substrate. The second is uncompetitive inhibition. That's where the inhibitor sort of competes with the enzyme substrate complex. And finally, mixed inhibition. Okay. So those are reversible types of inhibition. There's also irreversible inhibition, which we won't really talk too much about, because that in a sense really kills the enzyme. And you have, so we'll mention different types, or at least different examples of a couple examples of irreversible inhibition, but that's really just an inactivation of an enzyme, usually by covalent, in something like an inhibitor binding covalently to the active site. Okay. Right. You might also sometimes see inhibitors referred to as negative effectors. Okay, they bind to an enzyme and they decrease or inhibit that enzyme's activity, that enzyme's ability to catalyze the reaction. Okay, and again, three types: competitive, uncompetitive, and mixed. Mixed sometimes you see something that's called non-competitive, which is a simpler case of mixed inhibition. So that's a little bit confusing, right? But this uncompetitive is the one we'll primarily be focusing on. Okay, so first is competitive inhibition. Competitive inhibition is where you have an inhibitor, I, and it competes with the enzyme for that active site, right? So your enzyme can either react with the substrate and form that enzyme substrate complex, or it can combine with the inhibitor and form the enzyme inhibitor complex, which then does not catalyze the reaction. All right, this is really I think the simplest, easiest to understand. Okay, so this enzyme inhibitor complex won't bind a substrate and no reaction occurs. Okay, so that's why it's an inhibitor. So you have this enzyme, there's two equilibrium reactions competing here, and that's why we call it a competitive inhibitor, right? These two equilibria that are competing. Okay, the kinetics to this and the equilibrium, they get a little bit complex. Okay, so if we have our enzyme here, and it can either bind with our substrate, and you should think of these as double-headed arrows. It can bind substrate to form our ES complex, or it can bind inhibitor to form the EI complex. We can write at least this part of it, this equilibrium, in terms of our concentrations of enzyme inhibitor and enzyme inhibitor complex, and we get this equilibrium constant, capital Ki. Right, and then we can do some other things to it, right? The inversion and rearranging it. Okay, and you can kind of follow this math through if you're interested. But the main point here is we come up with this term alpha. Alpha equals 1 plus inhibitor concentration divided by our Ki. Okay, this term gives you an idea of the level of inhibition that's going on. Okay, so when you don't have an inhibitor around, so if this concentration here is 0, the alpha is 1. And when we add this to a Michaelis-Menten equation, we put our alpha value in the bottom multiplied by Km. Okay, so our alpha term goes into here, so Km times alpha. And this becomes our Michaelis-Menten equation for competitive inhibition. Okay. And we talk about the Km here, alpha times Km. We also sometimes refer to that as the Km apparent. Okay. There's no change in Vmax, and there can be an apparent increase in Km. Okay. Alpha is really a scaling factor. Removal of free enzyme from the reaction scales down the apparent Km value. All right. So here, alpha equals 1, that's no inhibition. So this would be our normal Michaelis-Menten curve when we're plotting V naught versus substrate concentration in green. As the inhibition increases, the inhibitor concentration increases, the alpha values will increase. As those increase, we're going to get an increasing of Km. So remember what that means. If Km increases, our Km value increases here, right? Our Vmax doesn't change. Vmax over 2 doesn't change. So if Km's increasing, that point where our curve touches Vmax over 2 is increasing down the x-axis, right? So the curve's really kind of just, I don't know if you could call this flattening out, I guess. It's not rising as sharply because they're still going to eventually hit the same Vmax, but it's taking longer to get there. So if we look at our, again, if we try to define, this is our Michaelis-Menten equation, that alpha for competitive inhibition, alpha is going to be multiplied by Km value. Remind ourselves alpha is 1 plus inhibitor concentration over Ki. Ki comes from this equilibria. It's our enzyme times inhibitor concentrations divided by enzyme inhibitor complex. Okay. What this does to our straight line plot is shown here, right? The Vmax doesn't change, so our intercept, y-intercept, 1 over Vmax, isn't going to change for these curves. Okay, alpha equals 1 is no inhibition. Okay, as alpha increases, right, our slope is our apparent Km, alpha times Km, divided by Vmax. So as alpha increases, our slope's going to increase, right? So the curves with higher slopes have more inhibition. And having a higher slope here, 1 over Km is going to get smaller. Okay. Next, we have what's known as uncompetitive inhibition. Okay, in uncompetitive inhibition, the inhibitor blocks the enzyme substrate complex. So that's after this equilibrium is formed. So there's no competing directly at that enzyme with the inhibitor with the substrate. They're not competing with each other. They act on different parts of the protein. Okay. And so you have an uncompetitive inhibition, you have the formation of ESI, the enzyme substrate and inhibitor. And to do this, again, we have a very similar term. We call this alpha prime, and it's 1 plus inhibitor concentration divided by Ki prime, right? So this is a little bit in blue here. These should be Ki primes, and that prime just means it's for uncompetitive inhibition. Okay, but in general, it's the same expression as for competitive. So in this case, our Vmax is going to be divided by that alpha prime. Our Km is going to be multiplied by it. What that does in reality, when you do the algebra on that, you get this expression where it's Vmax times substrate concentration divided by Km plus alpha prime times substrate concentration. So this is our altered Michaelis-Menten equation for uncompetitive inhibition. And when we look at our straight line plot for this, okay, what we have, no inhibition, no uncompetitive inhibition, we have this line. Okay. When we have uncompetitive inhibition, alpha prime is going to increase, right? So alpha prime will increase, that affects our intercept, but that doesn't affect our slope. It's not affecting Km over Vmax. Okay, so the slope of the line is going to stay the same. We're just changing our intercept, and our 1 over Km does change. Okay, so that's very important to remember. The Vmax and Km are actually both changing, but they're changing by the same amount. So the slopes aren't changing, but 1 over km, the x-intercept on this plot will change. Right, so you have a decrease in your Vmax, increase in km, and there you have a no change in km over Vmax. Right. Finally, mixed inhibitor or mixed inhibition, you have a part of the inhibition that's similar to competitive and part that's similar to uncompetitive, and you get enzyme being pulled out at two separate parts in the pathway. Right, and this gets very much more complicated. As you would expect, you might guess, our Michaelis-Menten equation for mixed inhibition, we have, as with competitive inhibition, we have alpha Km, the apparent Km, and we have alpha prime times substrate concentration here. So really this is a very complex system to draw out. But all we're doing is we're doing what we did for competitive and uncompetitive, and we're combining them in our Michaelis-Menten equation. Okay, what this looks like on our straight line plot is you get a little bit of both worlds. You get a change in the y-intercept and you get a change in the slope. And so what happens is you get this sort of look. So normally we'd have these plots in this node here would be along the y-axis. But now that's shifted. You can think of it as basically kind of just shifting over. Okay, so we have each of these plots as the inhibition increases, right? That the y-intercept is increasing, and as the inhibition increases, your 1 over km is decreasing. Okay, so you have a decrease in Vmax and a change in km. And as a summary, what do our Michaelis-Menten equations look like? Right, uninhibited, that's the normal situation. We have it with competitive inhibition, non-competitive, and uncompetitive. Right, probably better to look at these values here. So that's our summary for inhibition. And also important to kind of know how the straight line plots will change for different types of inhibition, because that would be a very easy question to ask on an exam, and it should be, if you remember them, they should be very easy to tell from those plots. Okay. Irreversible inhibition, okay, those are as I mentioned, things that tend to bind covalently to like a amino acid side chain in the active site. So in the case of some proteases, they have serine residues that are in the active site, and you can have molecules like this. This would be our inhibitor that binds covalently. So at this molecule, you don't really need to know this mechanism, but you have this fluorine group. That fluorine's lost, and you get the binding here of a very sort of stable covalent bond that's formed, and that inhibits the enzyme irreversibly because this can't be removed from the active site, and that this residue here, this serine residue, is catalytic. The enzyme needs it to function. And really this is the enzyme acetylcholine transferase, where you have acetylcholine, you take an acetyl group off of that, right? So here you have your serine, it grabs the acetyl group, choline leaves, and then it transfers the acetyl group to in this case water, to make acetate. right? So if you have this inhibitor that binds there, right, that binds irreversibly, you inhibit the enzyme forever. Okay, and this is acetylcholine is a neurotransmitter. So this inhibitor here, it actually a neurotoxin. It's used as an insecticide because it inhibits this enzyme that's needed in acetylcholine metabolism. Okay. Another example of irreversible inhibition or from molecules like penicillin, and they not really too important to know the exact mechanism, but they block a enzymes that hydrolyze that form the cross links in this beta lactam ring. So what that does, these beta lactam rings are important for the cell wall of a bacteria. So penicillin inhibits this enzyme that's needed to make these cell walls stronger, and the cell walls without that enzyme tend to weaken, and then the bacteria get very leaky and they're susceptible to bursting, and when they burst they die, right? Right, so finally we'll talk about well, I shouldn't say finally, we'll mention a little bit about some catalytic mechanisms of enzymes, and then at the very end we'll talk a little bit about regulation. Right, so there are four main types of catalytic mechanisms in enzymes that we're going to mention. First is acid-base catalysis. This is where enzymes give and take protons. Covalent catalysis is when you change a reaction path with a covalent bond to the enzyme. Metal ion catalysis uses metal ions as sort of redox cofactors. They're able to change pKa values. In that way, they're a little bit similar to acid-base catalysis, but they involve a metal ion. Okay, electrostatic catalysis. These are performing interactions like a charge interaction with a transition state. So a mechanism, I believe your book shows like chymotrypsin and gives examples of some of these mechanisms. We'll show a little bit some different examples maybe than the book has. Okay, the enzyme RNase A, it cuts RNA molecules, so chains of RNA. It can cut, and how it works is by acid-base catalysis. In the active site, this is our RNA chain, and in the active site there's a couple histidines. One histidine acts as a base, and it grabs a proton, and the other histidine that's protonated acts as an acid and gives up its proton. Okay. So what you have here is the arrows showing the mechanism, right? You have a grabbing of a proton. Those electrons attack this phosphorus. Then we have these electrons taking this proton from this histidine, and finally the electrons that were on this using this bond move down to that nitrogen. Okay, so that's our electron pushing, like we would do in an organic chemistry class. Okay, so that's our first step. What that does is it breaks this part, right? As these electrons grab that proton, this is now its own separate chain. It can leave. Right? We have this kind of a ring structure here formed by this phosphate, and then what that does is it then grabs that proton back that was grabbed by this histidine. Okay, it grabs that, and then a water molecule, this histidine can grab a proton from a water molecule, and then the OH group can then attack the phosphorus, and then that breaks, as we showed this, that's this step. Our second step, that breaks that ring here, and we get our phosphate group at the end. Now this part of the chain can leave. So that's an example of an acid-base catalysis. You have proton taking and giving in the enzyme. All right, covalent catalysis, it involves a transition state or transient covalent bond between the enzyme and the substrate. And you can think of it as changing a reaction pathway. So an uncatalyzed reaction would look like this. You have a A-B molecule that has an A part and a B part that they're connected by a bond, right? That could be cleaved by something like water in a hydrolysis reaction, and you get A plus B. To catalyze this, what would it look like? Well, you have that AB molecule still, but then you have plus X, and X would be an amino acid side chain in the active site of the enzyme, right? So AB plus that enzyme, then you'd form a covalent bond between the enzyme and part of the molecule, and the other part of that molecule would leave. And then the second step is the release of the part that's covalently bound to the enzyme. So in the second step, A is now leaving. Okay. This requires a nucleophile in the enzyme. That's what this X is. Okay. And these can be serines, thiolates, so like cysteine or methionine, amine groups, or carboxylic acid groups. Right. Metal ion catalysis, our enzyme carbonic anhydrase is an example of one of these. Right, we have in the active site, we have zinc 3 histidine residues that are bound to a zinc. And then the other coordination site on the zinc, a water molecule is bound. Right? That water molecule then becomes active because it's bound to this zinc, and it makes this one of the hydrogens on it easier to remove. It's removed by a histidine in the enzyme, and then you get this OH minus, and this is our activated form of water in that active site that can then react with our CO2 molecule to form HCO3, our bicarbonate. Okay, right. So this active site uses, you have that example of a histidine grabbing a proton, which is sort of like an acid-base catalysis, but it's really only one part of it, and that proton isn't used in the reaction itself. The resulting OH minus is what's used in the reaction, and that's because that zinc can activate the water. Right, so enzymatic reactions. Understanding the complete reaction mechanism of an enzyme requires a lot of things. It requires knowledge of all substrates, cofactors, products, and regulators of the enzyme. It requires knowing the temporal sequence in which enzyme-bound reaction intermediates form. It requires the structure of each intermediate and each transition state. It requires the rates of interconversion between intermediates and the structural relationship of the enzyme to each intermediate, and the energy contributed by all reacting and interacting groups in each step. Okay, so it requires a lot of knowledge to give a complete reaction mechanism of an enzyme. Currently, we don't really know all of these factors for even a single enzyme. So understanding reaction mechanism is really a very complicated situation. Okay, in gaining information of reaction mechanisms and enzymes can be a little bit tricky. I mean, luckily they do tend to follow the four primary types of mechanisms that we mentioned. Okay. Right. Lastly, we'll talk about regulation. Okay. There are four main types of regulation. One we've already seen examples of, allosteric control. That's binding of regulatory molecules at sites distinct and sometimes distant from the active site that trigger conformational changes that are then transmitted to the active site. Okay, allosteric proteins show cooperativity. Activity at one functional site affects the activity at others. And the example is hemoglobin that we've already talked about. Multiple forms of an enzyme is another way to regulate it. And when you have multiple forms of enzymes, we call these isozymes. Okay. These are homologous, so very similar enzymes within a single organism that catalyze the same reaction, but differ slightly in their structure and slightly in the kinetics of this catalysis. And they can also be regulated slightly differently. They can be targeted to different organelles within a cell or different tissues as well. Okay. Proteolytic activation. This is where you have hydrolysis of a few peptide bonds in a precursor to the enzyme, and once this hydrolysis occurs, then the resulting cleavage products are active. So you have a longer peptide chain, a protease cuts it at a certain point, and then what's the smaller piece becomes active. Okay. The precursor molecules are known as zymogen or proenzymes. Okay, so those are the longer inactive forms. Once the hydrolysis occurs, then we get our active enzyme. Okay. And we'll see examples of these. The chymotrypsin and also blood clotting is due to a cascade of proteolytic activation. Okay, and then finally we have reversible covalent modification, where you have the catalytic properties of enzymes are altered by covalent attachment groups, and the classic example here is of phosphate groups that can either activate the enzyme or inactivate it. Right. So allosteric activation or regulation we've already talked about, and that example is hemoglobin. Okay, so in an example, if we had no cooperativity, the curve here would look like this in the blue. When we have allosteric regulation of the enzyme, we get cooperativity, and it turns into this sigmoidal curve. Okay. Proteolytic activation. All right, the classic example here, some of these are shown, and these are all gastric and
pancreatic digestive enzymes, and they're expressed in zymogen form. So as they're expressed, they're not active, and that's really kind of important because you don't want these digestive enzymes cleave things like proteins, other proteins. So if you have these active within a cell, they'd be, eating cellular proteins that would be important. So you want to express them in an inactive form, the zymogen form. So in the case of the stomach, the active enzyme is called pepsin. The zymogen is pepsinogen. Okay. That's expressed. It is then excreted in the stomach, and it can be converted into the active form of pepsin. Okay. And the other example here is chymotrypsin. The active enzyme would be, chymotrypsinogen would be the zymogen, and chymotrypsin is the active enzyme, and that's a pancreatic enzyme. So if we have, using that example, chymotrypsinogen, the inactive form, so that's shown here. It's a 245 amino acid enzyme. That can be cleaved by another digestive enzyme, a proteolytic enzyme called trypsin. So it cuts peptide bonds, very specific peptide bonds. And when it does that, it cuts off the first 15 amino acids to give pi chymotrypsin. And then the chymotrypsin will then self-cut it, self-cleave, and form what's known as alpha chymotrypsin, which is the active. And you have three chains: the A chain, which is small, the B chain, which is the longer one, and then another smaller C chain over here. Right. And you see here shown some structural changes that occur and some important enzymes, amino acids in the enzyme. The positions change pretty drastically. Okay. Blood clotting involves a cascade of proteolytic enzymes. Okay, proteolytic enzymes, again, that means that enzyme cuts a protein bond, a peptide bond. Okay, and you don't need to know this. This is a little bit more complex than we need to get into, but you have these clotting factors that really they start as their zymogens, and they become cleaved, and by cleaving, factor 12 here becomes factor 12A, it cleaves factor 11, and so forth. You get this cascade of cleaving, which eventually gets to prothrombin to thrombin, and fibrinogen to fibrin, which then does the cross-linking fibrin clots. Okay, some compounds that act as anticoagulants, they end up blocking vitamin K, which is needed in this reaction. So you have a vitamin K dependent glutamyl carboxylase that cuts, and if you have some examples here are like warfarin is a classic anticoagulant. It looks very similar to vitamin K, but it's an inhibitor, so it inhibits this reaction and prevents this cascade from going further to the clotting. Okay. Right. Regulatory enzymes can also affect rates. So if you have an example of an enzyme C that has an active site that binds a substrate, sometimes you have another regulatory, either a separate enzyme or another part of this protein that's a regulatory site that can bind a certain molecule, and when it binds that effector molecule, it can change the binding characteristics of the catalytic site, right, and allow faster binding. So this is really sort of an example on the lines of allosteric regulation. Okay. And when we can look at the plots for some of these, okay, and if you have this plot would be our normal plot without any effectors on it. You can have an example of molecule that negatively affects the kinetics, the binding, right? So if the negative, that would increase the what we would call here we're calling it K.05, but it's very similar to the Km value. Right, and when it does that, that increases that value, so it pushes that out, so it sort of lowers how fast that's moving up to Vmax. Okay, and if you have the other example, if an activator binds, that would make the Km lower, right? So that would mean this curve is growing up faster. Right. You can also affect the Vmaxes as well. Okay, you can have a case where they're not affecting the Kms here, they're affecting the Vmax. And when you see that, if you have a molecule that negatively affects Vmax, it's just going to lower the Vmax. Okay, Km is staying the same in each case. And then a molecule that increases the Vmax, right, that would move it up higher. And actual example of this in phosphofructokinase, which is an enzyme involved in glycolysis. If we have here, this curve in the blue is without any effectors, and you notice that it looks pretty sigmoidal, so you can kind of tell there's probably some allosteric regulation going on here. When we have our inhibitor present, the curve looks like the curve here in the red. So that's our inhibitor ATP. Right? So what it's doing is that it's lowering the rate. So as we're following substrate concentration, that rate stays low when we have our allosteric inhibitor present. When we have our allosteric activator present, though ADP, our rate is really high initially with low substrate concentrations. Okay, and so the composite of the green and the red, what we get is this sort of the blue color here in the middle. Okay, effectors combine covalently, and they do this reversibly. And the classic example of that is phosphorylation. Okay, phosphate groups combine to amino acids that have OH groups, so tyrosine, serine, threonine, and also sometimes histidines. Okay, they combined and you get a phosphorylated enzyme. These can be positive or negative effectors of the rate. Okay. This is really complex and very, very much over our course, so you don't really need to know any of this. But if you're thinking about real-world situations, enzyme kinetics can get really complicated. And so a bi-substrate reaction, a case where you have an enzyme that binds two different substrates and makes one or two different products, are possible. And we get what's known as a ternary complex. Okay, so you have enzyme substrate 1, enzyme substrate 2, and then the enzyme substrate 1 and 2 complex. And that's what's needed to make your either one or two products. Okay. You can have this case as well where you don't have a ternary complex that's formed, and instead you have, the first substrate binds and you make a first product. That product leaves and a second substrate comes in and you make your second product. And this one is called either a ping pong mechanism or double displacement. I don't know, being in enzyme research, I've heard the term ping pong and seen those examples before. So you might run into that at some point, and that's what it's referring to. Okay, but again, this is way more complicated than we're going to get in this class. Okay, so the first one being the ping, and the second part being the pong. Okay. These are more realistic. You can have up to 3, 4, 5 substrates and cofactors that need to bind. Rate equations are going to get much more complicated, and again, way beyond the scope of this class. Okay.